Hi, in this video lecture, we're going to go through the rest of chapter 22, and here is a recap of what we saw before. So, do you remember which immune system? Was it adaptive or innate that was very specific? Meaning that will target very specific antigens or bacteria or viruses, right? It's your adaptive immunity work, for instance, through your T cells as well as B cells. So these are very specific type of immunity. Now, so our innate immunity, for instance, example, our skin, our tears, gastric juice, are non-specific. Now, the other difference between them is one of them has long-term memory. Do you remember which one has long-term memory? Right? Your adaptive one has memory, okay, because they will develop cells that keeps a long-term memory of what went on earlier. Now, our innate immunity has no memory, okay, no memory involved. So let's take a look at those type of invading agents. So who are these invading agents that get into your body and starts an immune response? So we call them antigens, okay? Or they actually came from antibody generator, the term. So they took out anti and they took out gen, making it into antigens. So what are the types of antigens that can start, for instance, an immune response? For instance, they could be made from proteins, they could be made from lipids, or for instance, carbohydrates, or nucleic acids. Okay. So these are, or a combination of them. Now, so for instance, these can be present in bacteria, in viruses, in the toxin that they produce, toxin, or sometimes it could simply be in egg whites and you start an immune response, or pollen, etc. So for instance, now imagine here is one antigen. Okay, could be a bacterium or a virus. Now on the surface of that antigen, here's these epitope. And that epitope could be made from protein, lipids, carbohydrates, or nucleic acids. So these epitopes, and for instance, on the surface of the antigen, you actually could have a different epitope. So one antigen, for instance, could have many different types of epitopes. Okay, and you might hear another word called antigenic determinants. It's the same meaning as epitope. Okay, so don't get confused by those words, even though it looks a little bit more fancy. Now, heptane refers to a smaller, for instance, substance, okay, that has one, what we call reactivity, meaning actually most of these antigens are going to have reactivity by interacting with your antibodies or the cells they come in contact with. So now for most general antigens, they will have both characteristics. They will have their reactivity, but they will also have their immunogenicity, meaning they can launch and start an immune response. Now, for our heptane, which are a little bit smaller, they have the reactivity, but they are not immunogenic. Okay, so for instance, unless they are bound to a much bigger molecule, an example, for instance, penicillin. So penicillin is a medicine, antibiotics. Now, so sometimes they can bind to a bigger protein in a patient's system and then causes become immunogenic. Okay. So this become very serious because that means some of these medicine can cause patients to develop allergic reactions to them. Now, who will recognize all these antigens in your body? We have a very clever system. We have these antigen-presenting cells, the APCs, and I want you to know them. There are B cells, our macrophages, as well as our dendritic cells. Okay. So they are responsible for recognizing them. Now, you probably will take a look at something and come across a phenomenon knowing that your immune system is very awesome because they can recognize and bind to many, many epitopes, at least 10 to the 9 power, that many. So how can they bind to that many, for instance, epitopes? For instance, you're going to take a look later on at a lot of protein receptors, okay, or protein molecules. Okay, this is an R that recognizes these antigens. Okay. And a lot of things actually came from a great degree of what we call genetic recombination. Okay. Your genes undergo a lot of shuffling in order to make many, many types of these protein receptors and protein molecules that recognizes these antigens. Yeah. So that's why even though we start from about close to 35,000 genes, but we can make, for instance, 10 to the 9 power of antigen receptors. That recognize a wide range of all these different epitopes. Now, so your antigen receptors, a lot of them located, for instance, on your T cells, okay, also in your immune cells. So they recognize that many antigens, as we said earlier, through a lot of genetic shufflings. So through genetic recombination. Now let's take a look at how your antigen presenting cells and presents these antigens. So over here, we're going to focus on those that are exogenous antigens, meaning that they are not from within the cells. These are, for instance, outside the body cells. So for instance, these type of antigens could be from sources of bacteria, viruses that didn't get into your cells yet, pollen, etc. Okay, so here is this big cell is your antigen presenting cell. And don't forget, this includes our B cells, macrophages, and as well as our dendritic cells. Okay. So what happened is, you have this exogenous antigen, for instance, that are outside your cell. Okay. So the first thing that will happen is they're going to be phagocytized, okay, undergo phagocytosis, okay, by your antigen presenting cell. They will engulf it, and then they are going to be present and fused with, for instance, lysosome. So over here, they are going to be degraded, or what you say, digested. Digestion into pieces, fragments. So basically, your antigen presenting cell takes it in those bacterium or viruses outside the body, outside the cell, and phagocytize it and then chops it into pieces. At the same time, they have a protein complex called MHC2. Now here is your major histocompatibility complex. And there are two types or two classes of them. This is class 2. So that's why AMHC2. AMHC2. So in your B cells, macrophages, as well as dendritic cells, they are made and they would be fused later on. The vesicles fuse with those parts of those fragments of the antigens. And then they bind to it, and so they form, for instance, a complex where you have what we call an antigen peptide binding to your MHC2 proteins. Okay. And then the vesicle are going to undergo displays right on your plasma membrane. So it will be inserted in the plasma membrane. Okay. Here is a very important thing is now they are ready to present this. Here is your MHC2 complex, and here is your antigen, which is chopped. Okay. So they are going to present them to other cells. Specifically, we'll see later on to a type of T cells, cytotoxic T cells, and tell the T cell to do the rest of the jobs. Okay. Now, on the opposite hand, here are your endogenous antigens. So, who recognize them? These are, for instance, those antigens that are inside the cells already. Okay. So, all bodies, for instance, cells are going to be able to do this. They will be able to, so any infected body cells, okay, in all regions of it, is they are going to, for instance, also digest these endogenous antigen. So these endogenous one, we say they are inside the cells already. For instance, we might have viral particle, viruses that already got in, virus that got into the cell and making copies and copies and copies of it already. So once inside the cell, they also will be chopped into fragments. And now watch out here, all your body cells, okay, all cells except red blood cells are going to make this MHC1 complex. So major histocompatibility complex 1. Okay. So then they will transport these major MHC1 molecule and they will bind to your antigens. And once they're all packaged, they're ready also to be inserted onto the plasma membrane. So they're also there to present it to, for instance, your cells, okay, ready for actions. Okay. Now, so let's take a look at the different types of adaptive immunity. So earlier we saw in previous lecture, that we went through the inborn types are innate immunity versus our adaptive ones. So now we took a look at the antigens and how they are presented. Now we're going to take a look at the cells that are doing the work. So cell-mediated one refers to your T cells, and antibody-mediated ones refers to your B cells. You'll hear another term for antibody-mediated, humoral immunity. Doesn't mean they're funny, but actually refers to they are present in a lot of our fluids, such as the antibodies that are produced by the B cells. So first, let's remind us, okay, where do these T cells and B cells come from? Do you remember where they are from? Right? They're actually from your pluripotent stem cells, and if you remember another word for it, your hemocytoblasts in your red bone marrow. So we have these pluripotent stem cells. Remember from before, they make other cell lines into red blood cells, platelets, etc. Today we're just going to focus on our T lymphoblasts, B lymphoblasts forming into what we call T lymphocytes, T cells, and B lymphocytes. Okay. Now, so they do come from your red bone marrow. Now, in order for them to become ready for work, for fighting on the battlefields, they have to go through training and training and training. Now, the first step that we'll go through is what we call activation. Okay, when they're just made, they're inactive, just like still undergoing a lot of training. But next, when they're activated, they will go through a second step called clonal selection. Okay. So they have to go through these two steps. Both of them can't miss one step in order to become ready for actions. Now, so let's take a look at the first step, activation. And here is quite complex, this diagram, but let's break it down. Okay. So here is our red bone marrow, right? Remember, they're in the spongy bone regions, in the pulcas. Here we have our stem cell that differentiate into what we call mature B cells. Okay, don't get fooled by it. Even though they're mature, they're not activated yet. Now our pre-T cells travels to another place. Where do you think they travel to? This was also another primary lymphatic organ, right? Thymus. Okay, so from the thymus, okay, they will make mature T cells. Okay, now that's why you see the T for thymus. Now B actually stands for bursa. When they first discover where they originate from was from the birds, bursa system, but in our equivalent one is our bone marrow. Okay, so same B for B, T for T. Okay. Now, so once they are mature, they travel to the second, what we call lymphatic tissues and organs. So for instance, in our spleen, our lymph nodes, lymphatic nodule, this is where that activation occurs. So let's first follow the fate of this one. Okay, mature T cells are going to differentiate into what we call inactive cytotoxic T cells. Now, we also have our inactive helper T cells. Okay? So they are very important. Two cell lines are made. Okay? So within your cytotoxic T cells, they make a very special surface protein called CD8. Okay? And if we compare that to your helper T cells, they make a CD4 protein on the surface. And this comes into play later on when they recognize different MHC complexes. Okay, let's follow first the fate of your helper T cells. Okay, in order to activate them, okay, they go through what we call recognition of other cells, for instance, through your antigen presenting cells, in order to form all these formation of their clone cells. Now, what they make are two types of cells. It would be active helper T cells, those that are doing the job, but the other set are your memory helper T cells, that they are going to remember a lot of things for a long time to come. Now, with the helper T cells, active helper T cells, now you can really help the other two lines. So they will help the cytotoxic T cells become activated, usually with the secretion of cytokines and etc., to form active cytotoxic T cells, as well as memory cytotoxic T cells. Okay. Now, same thing, the active cytotoxic T cells, now they're ready. They're going to go ahead and attack invading agents. Memory cells don't do much, but they will have their long-term memories. Okay. So Now let's take a look at the B cell line. So our B cells are the ones, for instance, having their antigen receptors on the surface. Now, they also will need the help of your um, active helper T cells. So in order to form into many clones. Now, the two type of cells that they form into, one is called plasma cell. These are the ones making these antibodies. Okay. So the plasma cells, your B cells that goes into plasma cell, they don't really directly attack the antigens, but they will have their ways to do so. Now the other line is memory B cells. Okay? And these will remember for a long time to come, and they will make memory necessary when needed. Now the antibodies, their action, we'll take a look at it in a moment too, but they will find their ways to inactivate antigens in the body fluid. Now one other distinction is your antibody-mediated one tends to direct against cellular pathogens that are outside the cell, whereas your cellular-mediated, using all your T cells, these are targeting intracellular ones. Okay? So over here, what I want you to know is where they start from, okay? And also the different types of T cells they branch into, okay? And the different types of T cells you end up with. Okay? Don't forget your active helper T cells, as well as the memory ones, and what your mature B cells form into plasma cells and memory B cells. Okay? All right, so let's take a look now at what if you have tons and tons of enemies. Okay? So think of these are your enemies, and these are your antigens with tons of epitopes on it. And you will have certain sets of antigen presenting cells. Yeah. So, but there's only you by yourself. You are an inactive helper T cell. You're not active yet. So sad. So what can you do? Right, there's so many of them. There's only one of you. Right? You have to make many more of yourself. So this is the second step of clonal selection. Okay? That second step actually occurs right around here for clonal selection, but I want to talk to you a little bit more on the first step, okay? The activation steps, okay? Because earlier we couldn't really see a closer look at it. So here is, for instance, how our T helper cells, which are inactive, are going to become activated, or active helper T cells. So to go through that first step, first is your antigen presenting cells, or B cells, macrophages, etc., dendritic cells. They present the chopped up antigen right over here. Okay, we're going to zoom in over here because there is a very important interaction between your antigen presenting cell as well as your inactive helper T cell. So if you zoom in over here, okay, there is a recognition, okay, between your MHC2 complex, okay, here, and your inactive helper T cell. The inactive helper T cell have proteins. First one over here is your T cell receptor, singular for now. So these T cell receptor is going to recognize the antigen, okay, that chopped out viral particle that was presented by MHC2 complex. Okay. So what we say is first thing that need to occur is antigen recognition. They have to be able to recognize that antigen, that foreign antigen. Okay. Now, secondly, that they have, so that's those first question mark over here. The second question mark over there is a co-stimulation has to occur. That means this CD, which is CD, which protein is that then? Is it CD8 or CD4? right, on helper T cell, it's always CD4, okay? So CD4 protein that anchors onto your plasma membrane of the inactive helper T cell also has to recognize your MHC2, okay? So this is your second question mark. So two things are needed for activation. Okay. First, you gotta recognize the antigen. Second, you have to both recognize your MHC2 complex. So in order for activation to occur, so now if they recognize everything, it's now what we call activated. And then it goes through clonal selection, meaning they will make copies and clones and clones and clones of itself. And they do make two different types of clones. That means differentiate into two different types. One is your active helper T cells. The other is your memory helper T cells. Okay. So very important message over here. Make sure you know what has to happen for activation, and you make sure you know which MHC complex, which CD4 proteins are involved, and what type of clones and differentiate type of cells they made. Now, so here are some of the cytokines. Now, let me digress a little bit, because from here, actually, to induce this clonal selection, you will need the help of a lot of cytokines. Okay, and for instance, the helper T cells, they will produce interleukin-2 and other ones to help signal proliferation and clonal selection of other cells. And here are the previous lists. So don't worry too much about it, but I just want to show you there's several different ones here. So specifically, interleukin-2 is secreted by helper T cells. Yeah, they will help you generate more clones of cytotoxic T cells, its own cell, B cells, and other cell types. Okay. Now, we also want to make sure you have tons and tons of clones of your cytotoxic T cell. So they also go through their activation as well as their clonal selection. So this time there is some differences. Okay, of course, things that needs to happen is they need to recognize the antigen that are presented by your infected body cells. So this time, not your APC cells, but infected body cells where the viral particles are replicating inside the cells. So what's happening between your inactive cytotoxic T cell as well as your infected cell is several recognition. Your infected body cells have these MHC1 complex. They presented the antigen also to your T cell receptors. Okay. This time, their CD protein is CD8, okay, not CD4. So these all have to occur. Your T cell have to make sure they recognize it. Oh, this is a typo. It should be cytotoxic T cell from your publisher. So this should be inactive cytotoxic T cell. And through antigen recognition, as well as co-stimulation with the help of helper T cell using cytokines, now the cell becomes activated. Okay, then they can make clones of themselves. So they will proliferate and differentiate into two types. One would be active cytotoxic T cell, the other one memory cells. So if you're the active cytotoxic T cell, you are going to attack infected body cells. Okay? And we'll see how they do those attacks. And your long-lived memory T cells will go on to carry on for years to come as well. So next, this is the battlefields. They're all activated. They make tons of clones of themselves. So how does your active cytotoxic T cell work? So here are your active cytotoxic T cell. It does two different things. Okay. So once they recognize your infected body cells, okay, here receptors. So they release granzymes. Okay? And granzymes, think of it as like protein that enzymes that digest proteins or proteases. So these proteases are going to chop up a lot of things in here where and causes your infected body cells to undergo programmed cell death, apoptosis. So your cell dies, and some of the microbes that are out will be phagocytized by other phagocytes. Okay, so they will kill the cells. Okay? All right. Now another way that they work through is to release porphyrin as well as granulysin. So porphyrin, which we'll focus on really, is a pore, a complex of proteins that they release from your activated cytotoxic T cells into your infected cells. What they make, imagine you have this plasma membrane and they make a hole in here. So here's outside of the cell, inside of the cell. So imagine if you have a hole, can things go in and out of this infected cells? Yeah, you bet. So that's why the cell is going to undergo cytolysis, lysis of cell, or pretty much it means it will burst. Okay, and microbes are going to be destroyed by granulysin and enzyme as well that works on it. Okay, so here is your action of activated cytotoxic T cell by literally killing the infected cells. Okay, make sure you know what they make. Definitely know your granzymes and actions of porphyrins. Okay. Now, our cells, immune cells, are very, very specific, and they can make sure our cells are doing all right. Now, if we have a change of normal cells that undergo transition into what we call cancerous cells. These cancerous cells will tend to actually project these tumor antigens. So these tumor antigens, in a way, then will be recognized by your T cells, okay, your lymphocytes. Okay. So once your T cells recognizes them, these tumor antigens, they can kill these cancerous cells. And a lot of time, your normal cells may have been infected with some viruses, and those are very specific cancer-causing viruses. So they are very effective when it comes to recognizing these antigens from cancer-causing viruses. But there are times when our T cell might overwork themselves because they're just simply doing their jobs. So, for instance, with organ transplantation, we see a lot of problem with this, for instance, with graft rejection. So imagine, for instance, you're getting a new liver, okay, from a donor, and on the surface of your new liver, they have their own specific antigens as well. But your T cells, yeah, as well as your other immune cells are going to recognize this as foreign. As a result, they are going to launch the attack onto this new organ because it is foreign to them, so causing graft rejections. And so it's very, very important that we do tissue typing, make sure they have a well-matched MHC complexes. Okay? Make sure this is as close as possible because each person are going to have their own unique MHCs. Okay? So you want to make sure it's very close together. And for patients who have organ transplant, because you want to avoid the T cells from killing these new organs. These patient has to be under very long-term immunosuppressant therapy. So, so you're trying to hide from your immune cells, but at the same time, these immunosuppressant that inhibits your immune cells will also put you at more risk for infections. Yeah. So there is a trade-off that you have to be very careful of. Now, how about our B cells? We talk a lot about our T cells, what they do to kill the cells, infected one. Now, B cell works a little bit differently. They are those that produce antibodies. So, our antibodies, for instance, they also that are produced from your B cells also undergo activation. That's the first step, and clonal selections. Okay. So look over here on the top, we have our inactive B cells, and they display their B cell receptor. Okay, just like your T cell has your T cell receptor. So they are the one who are going to recognize these microbes that are more exogenous on the outside of the body cells. Now they are inactive once they bind to the microbes or the antigens, and with the help of helper T cells. Okay, so then they become activated. Okay, so the helper T cell in this case helps with co-stimulation because they will produce interleukins or cytokines to help these become active. So now these activated T cells, okay, can undergo clonal selection. That's the second step. Okay, first step, second step. So they also make tons of themselves, but into two different classes. So one of the class is our plasma cell. We say they are the ones secreting antibodies, as well as the second class are our memory B cells that are long-lived. Okay, so for our helper T cells, for our cytotoxic T cells, as well as our B cells, they do all go through activation and clonal selections. Okay, cool, right? Now, here's a look of our antibodies. Okay, so actually, it's just one of them, or two of them. So a short name is AB, and another name for it, immunoglobulin. So you might also hear IG. And your antibodies or immunoglobulin really is made from amino acids. Okay. So, and then group into protein chains, yeah, or peptide chains. So if we take a look over here, there is 4 chains. Okay, they have each of these antibody has 2 light chains, 2 heavy chains, and let's take a look at where they are. So here is a light chain because they're much smaller. Here is another light chain. Okay, now the ones in the middle is your heavy chain. And here is the other set. Now all these chains, peptide chains, are linked together, for instance, by disulfide bonds. Okay, probably if you remember from your amino acids, we have our cysteine that has a sulfur group. So these are those disulfide bonds when they form into cysteine to another cysteine. Okay, so to hold them all together, now we also have these regions. Before I talk about antigen binding site, I want to talk to you about the V's and the C. So there are variable region on your on these chains, as well as constant regions. So when we talk about variable versus constant, these refer to the sequences of the amino acids. So where are some of these variable regions? They are over here in this stretch of it, as well as in this stretch. In the top stretch, B and B. So, and the other ones are the constant regions. So not much changes in these amino acid sequence, but definitely in the variable region. So which site do you think would be a good place to bind antigens? Would it be good to use the constant regions to recognize all different types of antigens, or is it better to use lots of various sequence to recognize various antigens? Right? You want to use the variable sites, and here are your antigen binding sites. Okay, here as well as here. All right, so make sure you can label and name these regions. Now here's five different classes of immunoglobulin. So you most likely see 80% of those immunoglobulin in your body are IgGs. They help you with everything. They're everywhere. Now IgA is more present, for instance, in our body fluids, okay, such as sweat, tears, saliva, breast milk, and GI tract secretions. So here is very important things. If you have a good cry, make sure you wipe your tears all over your face. Yeah, this also helps fights off any pathogens. Now IgM is the only one that forms into a pentamer. Okay, there are 5 unit ones. And IgG are found on some B cells, but in a very low amount of them. IgE has a very important reaction in allergic reactions. Okay. Now, so let's take a look now at some of the actions of your antibodies. So your book mentioned several of these. They can neutralize the antigen, or they can immobilize pathogens such as bacteria. They can also cause them to clump together, agglutination, and they can activate the complement pathway. What is this, right? We'll see it in a moment. And they enhances phagocytosis, which is really eating pathogens, cell eating. All right, so in the next slide, you'll see a generalization diagram. Now, over here, I want you to be able to recognize the antibodies as well as the antigen. They come together to form a very specific lock and key reaction. So this is a complex, an antigen, antibody complex. And they are very, very specific. Okay? Only a certain type of antibody is going to recognize a certain type of antigen because they are all made of different things, and only very specific one will recognize it. Once they form that complex, okay, they do several other things. They can inactivate, for instance, through what we call neutralization, okay, of the particles, the toxins. They can cause agglutination. Oops, missing a few words. So for instance, as we've seen, if a patient is getting the wrong red blood cells, okay, and they launch a clumping reaction. And they can also cause what we call immobilization of the viral particle. Now they also can launch the complement system, which are a cascade of proteins and to form into a pore. So as a result, things exit in and out of your cells, causing the cell to lyse and die. They also enhances inflammation, okay, as well as phagocytosis. So let's take a look. So here are, make sure you can name some of the actions of your antibodies. Here is a quick look on complement activation. Okay, so it's a cascade of events, okay, through a lot of proteins, okay, and you see C1 all the way to C9 depending on the order they are discovered. So basically, I don't want you to memorize all these things, but I want you to know what happens towards the end. But I will describe to you. So for instance, all of these proteins came from made from your liver. And inside the liver, once these, like a C3 protein, once it's split, it becomes activated. So for instance, the C3A can activate your inflammation response through the action of mast cells, and release of histamine from the mast cells will help with vasodilations, for instance. Now, the C3b particles help with phagocytosis and through opsonization, increasing the interaction of the pathogens to your phagocytes. Now, this then activate also C5, which breaks into two parts, okay? And C5a helps with the mast cell, but the rest are gonna go into a formation of this great big thing. So this I want you to know. So it forms into a membrane, a channel. So very similar to porphyrin. And once they form into a channel, things that are inside the cell can leak out. Okay, fluids from outside, of course, can leak in as well, then causing your cell to lyse, so cytolysis. Okay. So here's the action of complement activation that can be activated or enhanced by your antibodies as well. And last few slides, we're going to take a look at immunological memory. So we'll focus on down here. These are the times through days. On this side is the number of antibodies that are in the cells in your body. So let's take a look. Follow the IgG line, okay, the blue line. So for instance, if you got sick, okay, you were exposed to a cold virus, not knowingly, and for instance, then it takes a while for you to develop symptoms, etc. But inside your cells, your B cells are working. So they are the one making your IgM as well as your IgG antibodies. So you will see a first lump. These are your primary responses after you're exposed. But let's say you all got better, but you have a second exposure to the same cold virus. Not again, but you did have it again. But next time you see it again, your IgG levels are going to go all the way up. And this is because all your plasma B cells remembers, okay, as well as your memory, specifically your memory B cells remembers what's going on. And plus the action of your plasma B cells, you're making tons of IgG this time to combat that same covirus. So you'll fight it right away. Now, so ways to acquire the adaptive immunity, we can do it naturally or artificially. And there are two ways to do it naturally. You can do it actively. That means, so you are exposed to a microbe. Yeah, you really got the pathogens from someone else and you're developing the antibodies on your own. Or we can do it passively through one very good example of, for instance, through breastfeeding. Through breast milk, moms can actually transfer IgG antibodies to the fetus. Okay? So it's very important, if you could, and to offer breast milk to your little ones for at least 6 months after they are born to help them fight off the pathogens. Now we can also do this artificially. So how can we do this artificially? Through active immunity, right? Instead of naturally getting sick, you are artificially getting sick in a way, but through vaccination. So, but these type of vaccines are weakened type of pathogens, or so weakened viral particles. Okay. So they are, for instance, immunogenic, but they are not pathogenic. So they should not make you very, very sick. Now, another way to artificially acquire these, we can do it passively. So, for instance, after a snake bite, you may be given the serum that already contains the antibodies already. And so these are intravenous injection, but this is short-lived, yeah? You won't be able to, you might have to do this again if you've got another snake bite. So here are some of these important aspects of our memory. Now the last thing is going to be about recognition of self versus foreign. Now our cells that we say, using our T cell as an example, we say only 2% of them survive in the thymus. And it's because they have to be very well selected. They have to undergo self-recognition, meaning that, for instance, the T cell receptor has to be able to recognize the MHC complex, the corresponding ones. And also they have to have their specific CD, either for instance 8 for cytotoxic T cell or cytotoxic for helper T cell would be CD4. So they first have to be able to recognize these MHC complexes through what we call positive selection. So they have to be able to make sure they can recognize them. Okay. Now the second selection is they have to be able to self-tolerate their own. For instance, self-tolerance means they actually have to be able to recognize not their own antigens. Okay. These cannot be self-antigens. Okay. So if they recognize self-antigens, they will die, undergo apoptosis. So this is what we call a negative selection, because only those that doesn't recognize the self-antigen will survive. So let's take a look at the next slide. Okay, we'll focus on just the T cells, okay, in this case. And so does your immature T cell, these are still in the thymus, okay? And will they recognize your self MHC proteins? If they don't, they die, okay? Yeah, because they have to recognize it. So it's through a positive selection for those, if they recognize the MHC proteins, they survive. Now the second step, okay, is, are they be able to recognize self peptides? If they do, they will die also. Okay, and some they do, but they undergo what we call inactivation. They don't die, okay, but they are just hanging around. So they are not able to start an immune response. So only those T cells where their T cell receptor are not able to recognize self-antigen through a negative selection, then they will survive. So that's why we say about 1 to 5%, yeah, of them really survives. That they will recognize their own self-MHC protein, but not self-peptides. Okay? And now, if once they're on the battlefields, okay, they are in the lymphatic tissue, so they will be able to recognize the antigen, and with co-stimulation, yeah, then they will undergo clonal selection. Yeah. Right. So make sure you can follow through and know what it means to undergo a positive selection versus a negative selection. Okay. And as we undergo a lot of stresses in our daily life, they have a funny way of inhibiting our immunity. So it's very important. Make sure you find a way to release your stress. Could be from this class, could be from me, could be from studying. So for instance, I also have my own stress, but I will definitely have a way to release the stress. And I love my Korean dramas. When I see them, it feels like, oh, my troubles are not as bad as theirs. Now, as we age, many things changes too. We have less reactive responses to vaccines, and we tend to have maybe development of auto-antibodies that recognize self-antigen and causing autoimmune diseases. So this is also very drastic. Now our T cells, our B cells also tend to be less responsive as well as we get older. So take good care of yourself starting now. It's never too late, yeah, to always make sure you have or your immunity all geared up to help you fight pathogens. Now, the last slide I want to talk to you about is a type of virus. It's HIV virus. Is our human immunodeficiency virus. So I hope I didn't misspell it. Human immunodeficiency virus. Now, this is the virus that causes AIDS, or what we call the acquired immunodeficiency syndrome. So first, let's take a look at a diagram depiction of HIV. So we say these viral particles are really not living, but once they get into a host, they become alive. Now, they also have their own sets of genetic material. In this case, HIV has RNA as their genetic material. And inside, they also have viral enzymes. Okay. For instance, reverse transcriptase is one. And also there's also other protease, integrase, etc. Now, once, for instance, these cells target or gets into your system or anyone's system, what it does is they target a very specific type of T cells. They actually target the helper T cells. Now, by binding through these little knobs to make a match, and then they enter into these helper T cells. So if your helper T cells are affected, now what do you think happened to your cytotoxic T cells as well as your B cells? Right, they cannot become activated, so you have less of them to help you fight pathogens. So as a result, infection occurs. Okay, grand scale of them. So there are treatments and medicines that have been developed to fight against these viruses. One thing, because once they get into a host cell, these RNA cannot be read. They actually have to be transcribed again from RNA back to DNA. So that's why we call it the enzyme, the reverse transcriptase. So as a result, treatments is to develop inhibitors, medicines, drugs that are inhibiting the reverse transcriptase, so your RNA cannot be transcribed back into DNA and affect the host cell. There are also inhibitors for proteases so that they can't chop down some of the proteins and make them active. Okay? So it's very, very important to know how it works. And some other viruses, instead of having RNA as their genetic makeup, they have DNA, so that they even skip a step to do so. But it's always good to be very, very careful and to know your surroundings and avoid these type of viruses. Now, okay, this is the end of chapter 22. We'll see you in class or in another video lecture.